So good morning or good afternoon. My name is Stephane Boissel. I'm the CEO of SparingVision, and it's my pleasure to be presenting SparingVision today. SparingVision was created to disrupt the field of genomic medicine in ophthalmology. We are developing the first mutation-agnostic gene therapy in the eye to treat RP patients. We believe that the field today is mostly about using the same recipes to go after the same target, and that first mutation-agnostic product will dramatically change the landscape for genomic medicine in ophthalmology. If you are familiar with RP disease, you know that it's a slowly evolving disease that starts with a mutation in the rods for most of them, and progressively with this genetic mutation in your rods, you start to lose this set of photoreceptors, and the disease evolve progressively so that at one point you also start to lose your cones, and eventually you turn blind. And rods are responsible for peripheral or night vision, while cones are responsible for central vision and color vision. So you can live with rods, but without rods, excuse me, but you cannot live without cones. And most genetic mutation in this disease affects primarily the rods, and most gene therapy being developed in the space will go after replacing missing or defaulting genes in the rods. The problem with this approach is that you have something like 70 different genes and 200 different mutations potentially affecting the rods. And so it will take a lot of time and a lot of effort and a lot of investment to cover that whole spectrum. And Luxturna, the first ever approved gene therapy in the space, is a good example of that fact, given the percentage of RP patient it is potentially addressed, which is in the 2 to 3% range. And with a mutation-agnostic approach, the idea would be to treat patients independently of what has caused their disease to start with. So as you can see from the slide, the competitive landscape, most of the players are in stage 1, trying to again replace a missing or defaulting gene. And you also see some technologies such as autogenetics or stem cell or transplantation or some cellular therapy approach being developed for the most advanced stage of the disease. But really, there is nothing in between stage 1 and stage 4. And this is where most of the patients are diagnosed. Unless you have a clearly identified genetic default when you are born, usually you are diagnosed when you are in, let's say, the 20s or 30s, when you have already lost some, if not all, of your rods, and so your peripheral vision has been damaged. And at that time, there is no treatment. I mean, replacing a missing gene or a defaulting gene in a rod that no longer exists is just not possible. And so the idea of our lead product, SPVN-06, is to come at the point where most patients are diagnosed, stage 2, and independently of what has caused the disease, to prevent the cones from degenerating after the rods have already degenerated. And the whole idea was started 20 years ago or so by the group led by José Sael. At that time, he was leading the Institut La Vision in Paris. He is now chair of the ophthalmology department at the University of Pittsburgh. And he tried to understand in the RP disease why, given the fact that most mutations affect the rods and not the cones, eventually the cones were also dying. And again, this is what is leading to blindness. He had a few hypotheses, and one of the hypotheses that he later confirmed and that he later deciphered the mechanism of action of is the following. There is in the rods a factory to supply the cones with a neurotrophic factor, which will be totally critical for their survival. And that factory, or that neurotrophic factor, which is called RDCVF, which is short for Rhone-derived cone viability factor, is again produced only by the rods and associated with the other exon from the same gene, the gene being NXNL1, that was encoded by the two scientific founders of the company, including José Saël. So RDCVF associated with the longer form or the longer exon of the same gene, RDCVFL, which is a very potent antioxidant. It's an enzyme that will protect the cones against oxidative stress, while RDCVF will favor the supply or the entry of glucose into the cones. The combination of those two factors, again, two isoform or two exons of the same gene, will be a very potent protection for the cones while the rods are gone. And the product FBN06 is really the combination of those two isoforms from the same gene into an AAV vector, with two very specific promoters. One specific for RDCVF, which is an ubiquitous promoter that will lead to the expression of RDCVF, again, the neurotrophic factor part of the product, into the RPE in the absence of rods, while RDCVFL, the long form, which is again the antioxidant enzyme, will be associated with a cone-specific promoter that will lead to the expression of this part of the product, specifically into the cones. So over the last few years, many different proof of concept have been published in various models, being rodent, mice, and rats. And as you can see from this slide, the impact of the product, both in terms of the function, but also in terms of the morphology of the cones and of the retina, was very spectacular. And more recently, we have started to test the product in an experimental model of pigs expressing the rhodopsin mutation. The model is called a P23H transgenic pig. And as you can see from the left hand side of this slide, there is a very significant impact of SPVN06, the product, injected into a treated animal compared to the eye of the same animal not receiving the treatment. And as you can see from the slide, we measure to characterize this impact on the morphology of the cone. We measure the length of the outer and inner segment of the cones, but we also measure the presence of hemopsin in the cones of the treated eye. And this is not shown here because it's not yet published. It will be communicated at the upcoming ARVO meeting, but we've seen some very interesting quantification of that impact. On the right-hand side of the slide, there is a snapshot of the result of the first safety study that we've conducted in NHP. That was a pilot study that we decided to do ahead of the 3 months pivotal tox study in NHP, which is currently ongoing. And from that first study that was conducted in 2020, we can see that we have a very clean safety profile. Of course, this will have to be repeated in a larger number of animals over a longer period of time, and this is exactly what we are currently doing. The pivotal tox study is really the last study between us on IND submission, which we expect to do in Q3 of 2021, the goal being to start a first-in-man trial by the end of the year. So where are we today? In terms of manufacturing, we've recently filed the product. It's ready to go. We are going to manufacture a second GMP batch in 2021 to be used as a backup to the first GMP batch. And importantly, we have started to work on the commercial process development. What we are currently using is a process that is good enough to go into a first-in-man study, but we will definitely need to improve the process in order to launch the product. And this is something that we've decided to do early enough so as to plan for success. In terms of IND enabling studies, I've already mentioned that the last thing between us on IND filing is really the pivotal tox study in NHP, which is currently ongoing, and so far so good. In terms of regulatory and clinical development, we have received feedback from both FDA and EMA. FDA through the pre-IND meeting process, and EMA through the protocol assistance process. And on both fronts, we are good to go. They have validated the CMC plan, the preclinical development plan, and the clinical development plan when it comes to the first-in-man study. We have conducted we are conducting two natural history studies, one retrospective. We are just finalizing the study report, and one prospective study. Of those two studies are going to help us further define the clinical endpoint, but also characterize or better characterize the patient population that will be most suitable for that kind of treatment. And when it comes to the prospective study, the idea would be to transfer some of the patients in the prospective study in the next-in-man study, not the first-in-man study, but the study that will come next, and that, if everything goes well, could be a pivotal or registrational study. We have designed the phase 1 study, which we expect to be starting the enrollment on by the end of 2021. That will be a typical safety study with dose escalation, 3 doses on 5 different cohorts based on different level of severity of the disease. We've recently closed our Series A, on the second part of our Series A, 60 million euro. That gives us enough cash runway to go up until the end of 2023. We are currently embarking into an aggressive plan to refinance the company so as to extend that cash runway, not only to finance SPVN06, but also to finance the other development in the pipeline. We have put together a team of experts in the field. The last 3 additions were Florence Lorget. She's based in San Francisco. She heads development science for SparingVision, and she was previously with me at Sangamo, and prior to that, she was working in the ophthalmology department at Genentech. Rajiv Gangurde, who is based in Boston, is our CTO. He came from Voyager. And more recently, Dan Chung, who joined us as CMO. He was previously with Spark, where in the last 7 years, he has led the development and the launch of Luxturna. And again, on this slide, on the right side of the slide, you see the very short bio of the 2 founders, José Cel and Thierry Leveillard. And as I said earlier, José Cel, who is still very much involved in the story, is currently the chair of the ophthalmology department at the University of Pittsburgh Medical Center. So this is the last slide of my presentation. These 4 boxes are really expressing what is driving us. We are an ophthalmology genomic medicine play. Ophthalmology is the only TA we are working on. Genomic medicines are the only technologies we are developing. Our lead product is a gene-independent approach. This is the first of that kind that will be tested in man, hopefully in 2021. We are driven by scientific excellence, and the team is focused on just one thing, is to execute so as to bring that new kind of medicine as rapidly as possible to the patient. And with that, I will conclude my presentation for today, and I thank you very much for you to be attending this presentation. Bye-bye.